welcome to my channel. As you all know that in my previous video I have discussed about lifespan, reproduction, difference between the asexual and the sexual reproduction, and I have mentioned the names of different types of asexual reproduction. Just in case you have missed my previous video, you can watch it through the link I have mentioned in the description below. So in today's video I will explain fission and budding in detail, right? As I've already told you, there are 6 important and different types of asexual reproductions we have. So let's start with the fission. The first type of asexual reproduction is fission. By fission we mean to split or to divide into something. Fission is of 2 types further. We have binary fission and multiple fission. Under binary fission, we will be talking about 3 types further. That is irregular binary fission, transverse binary fission, and longitudinal binary fission. Okay. In irregular binary fission, I'll be explaining with the help of an example of amoeba. Under transverse, we'll be taking the example of paramecium. And in longitudinal, we'll pick up euglena. Okay. And then we'll talk about the multiple fission in amoeba only in detail. Right? So, moving on to the first one, irregular binary fission. Now, first of all, binary means 2 and fission means to split. Right? Binary means 2 and fission means to split. When something is splitting or getting divided into 2 parts, then we call it as binary fission. Right? So binary fission is basically defined as the process in which parent organism divide itself into 2 daughter cells or 2 daughter organisms. Right? So in the binary fission, the single parent is going to divide itself on maturity into 2 organisms. The next generation will be having 2 organisms, right? And both of those organisms will be identical to each other and to the parent. So let's take an example. In the case of amoeba, what happens is, suppose this is the parent amoeba having irregular shape, as you can see, that is cell membrane with the nucleus at the center and lot of cytoplasm. Right? Now this parent amoeba when it grows and gets mature, it will first replicate its nuclear content. Whatever is there inside the nucleus of the parent amoeba, that will be replicated. Okay, so that will be getting doubled. Fine. So it is actually preparing itself to undergo reproduction. So as a result of that, the size of the nucleus increases. The size of the nucleus increases. So when the size of the nucleus increases, after this, the nucleus elongates. The nucleus of the parent amoeba elongates, right? So this is the elongated nucleus. Right? This nucleus, after the elongation, undergoes nuclear division. A constriction appears in the middle of the nucleus, somewhere here. A cut or a constriction appears in the middle of the nucleus. As a result, this nucleus gets divided into the two daughter nuclei. So we'll be having this type of structure now. There will be two daughter nuclei. Clear? Now what will happen after this? As we know, there is a lot of cytoplasm inside. After the division of the nucleus, which is known as karyokinesis, splitting of the nucleus into the two daughter nuclei is known as karyokinesis. Karyo means nucleus, right? So after karyokinesis, it is further followed by another process there's a sort of imagination start appearing in the cell membrane, and slowly the constriction appears which separates the single parent amoeba into two daughter cells or daughter amoebae. So this breakdown of the cytoplasm, it is known as cytokinesis. After the cytokinesis, what will be the result? We'll be having two daughter amoebae. With enough amount of genetic material that is in the nucleus and lot of or equal amount of cytoplasm, right? So these two are this and this. These two are daughter cells or daughter amoebae. Clear? So let me just repeat what happened here is this parental amoeba is having the nucleus. Once it gets mature after the growth, and once it is ready for the reproduction, the nucleus of the amoeba replicates everything. The genetic material gets doubled. So when its size of the nucleus increases, after that the nucleus elongates. Due to the elongation of the nucleus, a constriction appears in the middle, somewhere in the middle. This constriction divides the single nucleus of the parent into two daughter nuclei. So once the daughter nuclei are formed, which is known as karyokinesis, these are the two daughter nuclei, karyokinesis happens, then the cytoplasmic constriction start dividing the parent amoeba into two daughter amoebae. And this breakdown or breaking up of the or splitting of the cytoplasm is known as cytokinesis. That results in the formation of two daughter cells which are identical to each other and also to the parent amoeba. Clear? So let's talk about the features of binary fission. The important features are First of all, parent give rise to two daughter cells in binary fission. Parent divides into two daughter cells. Then karyokinesis as well as, that is, splitting of nucleus. As well as cytokinesis takes place. Which is splitting of cytoplasm. Right? So both karyokinesis and cytokinesis takes place during the binary fission. Now, what is the nature of these organisms which are formed? Are they similar to the parents? Yes. So the daughter cells are, or daughter organism, are identical to each other as well as to parents. Right? So basically the most common type of cell division which takes place during binary fission is mitosis. Mitosis is the cell division which is involved. Now the very important point, why are we using this term irregular in case of amoeba? Because when amoeba divides, there is no specific plane of division. The amoeba can divide from any particular part of the body. The cut can appear from any side. Okay? So there is no specific, it's not longitudinal or it's not transverse or vertical or horizontal. There is no regular plane of division in the case of amoeba. This cut can appear from anywhere. It can appear from here, can appear from here, can appear from here as well. Right? So that's why it is an irregular binary division. Right? Irregular means there is no specific plane of division. There is no specific plane of division in the case of irregular binary fission in amoeba. Clear? Then as you can see, by the end of the process, no parental entity is left. There is no parental part which is left. So next feature is that no parental part is left after the cell division. As it is clearly visible that by the end of the process, once the daughter amoeba are formed, there is no parental entity or a parental part is left. Right? The entire parent got converted into the two daughter cells. This process of binary fission is specifically carried out during favorable conditions. And this is the only way of reproduction in the case of all the prokaryotes and in few eukaryotes. Only way of or mode of reproduction in prokaryotes. Prokaryotes as in bacteria. And few eukaryotes. So all the prokaryotes, they divide by binary fission only, right? And few eukaryotes, they also undergoes the process of binary fission. So this is about the binary fission in detail, specifically the irregular binary fission. Now we are going to talk about the transverse and longitudinal binary fission. Though there is not much of differences between the irregular binary fission and transverse and longitudinal binary fission. The concept is almost same. There is only one difference, that is of plane of division. In the transverse division, the plane of division is transverse, and in the longitudinal, it is along the longitudinal axis of the body. Rest all the features of binary fission in amoeba, paramecium and euglena are exactly same, right? So, on the basis of plane of division, we have already seen that amoeba undergoes irregular binary fission. Which I've already explained, right? Now let's talk about paramecium, which undergoes transverse binary fission. Right? Now what happens here is, first let me draw the structure of paramecium. As it has a slipper shaped structure, the entire body is covered by pellicle, a soft flexible layer, that is pellicle, and it is further comprises of large number of hairy projections, which are nothing but cilia, right? So somewhere towards this end we have an oral groove and cytopharynx and reservoir where the food vacuoles are formed. So the entire paramecium is filled with cytoplasm. There are two nucleuses in the paramecium. Macronucleus, the large one, and the small one is micronucleus. Alright, this is the oral groove. This is pellicle, the outer layer. These are cilia, as you can see. This is macro or meganucleus, and this is micronucleus. Now what's going to happen when paramecium is ready to undergo binary fission? Both the nucleus of paramecium elongates, they duplicate their content and they elongate in size, they increase their size, and the oral groove also duplicates itself. Initially, the oral grooves start disappearing and the two nuclei, they get elongated like this. They have duplicated their content, the genetic material, and they are ready to split, divide into two daughter nuclei, right? Now, both the nuclei, they got split into the two, and this oral groove also gets duplicated. So we'll be able to see this kind of structure now. This is kind of like one paramecium, and this one is the second paramecium, right? And they both have their own oral groove. They both have their nucleus, meganucleus and or micronucleus and micronucleus individually, right? So this is the transverse plane of division like this. They will get cut or get separated from each other through the transverse plane of division, right? That's why this is known as transverse binary fission, because at the end what we are going to get, we will be getting the 2 paramecium. So it is ultimately binary fission only, but the only difference is the plane of division. Like in amoeba, there was no specific plane of division, but here the plane of division will be transverse. They will be separated transversely and they'll get separate like this, and the new organisms will be formed, right? This is about the paramecium, transverse binary fission. So now third type of fission, binary fission, is the longitudinal binary fission, which is performed by euglena. It carries out longitudinal binary fission. Now let's draw the structure of euglena first. It has elongated body, looks like a leaf. So somewhere here, there is a small structure which acts like a photoreceptor. This structure is known as stigma. Okay, there is a outer covering of, that is again plasma membrane is there, and you can see this bulging structure. This opening is actually known as cytostome, right? Then there is a neck-like structure. This is known as cytopharynx. And this bulging portion is known as reservoir. Inside the reservoir, there are present 2 flagella, a short flagella and a long flagella. So, and longer flagella comes out like this. Okay, this is a flagellum. Okay, these are arising from these basal granules. Then there is a centrally located, somewhat centrally located, large size nucleus. This is nucleus, okay, and large number of green colored chloroplast. Now what will happen here is, this is going to divide longitudinally, means lengthwise. This was widthwise, transverse. This is lengthwise. So when it has to divide lengthwise, what happens is, initially the flagellum and the cytostome, cytopharynx and reservoir, this part gets splits up, gets duplicated, right? And it looks like this. This is one euglena and this one is second euglena. Rest of the body is still intact. Right? So it's somewhat like this. This is again flagella got duplicated, right? The cytostome part is also got duplicated. So you can see this portion, this kind of invagination, it start moving or going downward, and a constriction is going to appear, or a cut going to appear from here towards this side, right? So now what will happen here is, these 2 euglena ultimately will get separated like this. Slowly and slowly the cut gets completed up till here, and we'll be having 2 euglena. So ultimately each of them will be having their nucleus, each of them will be having their chloroplast, each of them will be having all the required structures a particular parental euglena comprises of, right? So ultimately these two will be separated lengthwise, they'll cut and we'll get 2 euglena. That's why the plane of division is along the longitudinal axis. That's why it is known as longitudinal binary fission in euglena, right? So this is about the type of binary fission. Now we'll move on to the next type of fission, that is multiple fission. Now moving on to the multiple fission, this is the second type of fission which is mainly carried out by the organisms during unfavorable conditions. Like when the conditions are not good enough to carry out the normal binary fission, then organisms switch on to the multiple fission. So we are going to take up the example of amoeba here. Amoeba do undergoes multiple fission as well under unfavorable condition. Now let's see what happens. As we know that amoeba, it's a single celled unicellular organism and it has these projections outside which are known as pseudopodia. You know, these are the false feet, pseudopodia. Now when the condition becomes unfavorable, environmental condition, during that time what happens is amoeba withdraws its pseudopodia completely and convert itself into a circular or a ball-like structure. It withdraws its pseudopodia and acquires this particular type of structure. Now as the conditions are not good outside, so in order to protect itself, amoeba is going to secrete some protective layers or envelopes around it. They are 3 in number. So it's going to secrete 3 layers or 3-layered structure, right? These 3 layers are collectively known as cyst wall. So amoeba secretes these 3 protective envelopes or layers which are collectively known as cyst wall, and these layers, they protect the amoeba during unfavorable condition. So now what will happen? On return of favorable condition, amoeba undergoes multiple fission. Let's see what happens. The nucleus of amoeba, parent amoeba, it undergoes multiple division. As a result, large number of daughter nuclei are created. And let's see how. So this nucleus will undergo multiple division and it produces large number of daughter nuclei. Right? So these are daughter nuclei. Don't forget that everything is happening inside the cyst wall. This structure is also having the covering which is known as cyst wall, which is 3-layered. I have already told you about that. So the cyst wall is still there. It is still there. Though the conditions are favorable, but cyst wall is still present. As we know that inside amoeba there is a lot of cytoplasm present. Now what will happen to these daughter nuclei? Each of the daughter nuclei gets covered by some amount of cytoplasm and a membrane, right? Each daughter nuclei gets enveloped by a membrane and it has a lot of cytoplasm individually. So cytoplasm gathers around and the membrane is created. So can we say that now each of the daughter nuclei is looking like a miniature amoeba, right? So in this way we have large number of daughter cells. There will be a cyst wall, and these daughter cells, each of the daughter cell having nucleus, its own nucleus, cytoplasm and membrane, these are known as pseudopodiospores. Pseudopodiospores, right? Now what happens after this is, as the conditions are favorable, after some time when these pseudopodiospores are grown enough, the cyst wall ruptures, it breaks down. It's like this. Cyst opens up and all the pseudopodiospores they start moving out of the cyst wall, the broken ruptured cyst wall. So all the pseudopodiospores, they come out of the cyst wall, they move out of the parent organism, they are released out. So pseudopodiospores are released. They are released out when cyst ruptures. Right? So what happens here is, first of all, this is a parent amoeba and the conditions are not good for amoeba to carry out the binary fission. So under the unfavorable condition, it is going to secrete a cyst wall, 3-layered envelope around it to protect itself from the outer conditions. So the cyst wall protects the amoeba, parent amoeba. Soon when the conditions become comparatively favorable, the nucleus of a parent amoeba undergoes division multiple number of times and create a large number of copies of nucleus. So a large number of daughter nuclei are created here, right? Now slowly each of the daughter nuclei gets covered by some amount of cytoplasm and a membrane, right? Individually, now the structure is known as pseudopodiospores. They have a spore-like structure, right? So once the conditions remain favorable, the cyst breaks up, this cyst ruptures, and once the cyst ruptures, all the pseudopodiospores, they start moving out. each of the pseudopodiospore grows or develops into young amoebae. Right? So this is about the multiple fission. Fine. Now there are two terms which are very important when we talk about multiple fission in amoeba. First of all, when there is a formation of cyst wall takes place, the formation of cyst wall, this process, cyst wall formation, this process is known as encystation. So this term is very, very important. Formation of the cyst wall is known as encystation or encystment, right? Secondly, the release of pseudopodiospores from the ruptured cyst. This process is known as sporulation. So when we talk about multiple fission in amoeba, we need to keep in our mind the two important terms: encystation, that is the formation of cyst wall during unfavorable condition, and when the pseudopodias are released out of the ruptured cyst after performing the multiple fission, then this process is known as sporulation, right? Because the spore kind of structures are released out of the ruptured cyst. Yeah. So this is about multiple fission in amoeba. So what are the features of multiple fission? It is a type of asexual reproduction in which a parent organism gives rise to many daughter cells. So parent give rise to multiple daughter cells, right? That's why it is known as multiple. Multiple means many. So many organisms are produced when the parent organism splits up. Okay, so parent give rise to multiple daughter cells. This process specifically takes place during unfavorable condition. Right? When the process takes place in amoeba, there will be two important processes which are included. One is encystation, that is the cyst wall formation, and second one is sporulation, that is release of pseudopodiospores when cyst wall ruptures. Right? In this process also, as we have already seen that once the pseudopodiospores are released out of the cyst wall, no parental part is left. Okay? So parent body did not exist at the end. Right? So these are some of the important features of multiple fission. Multiple fission is carried out by other organisms as well, like Plasmodium. As you know that Plasmodium is the causative agent of malaria, and it is also a protozoa. So Plasmodium also undergoes multiple fission. This multiple fission is a normal mode of reproduction in the case of plasmodium. So this is about the features of multiple fission. Now we are going to talk about budding. Now the second type of asexual reproduction is budding. In budding we are going to talk about two types, that is external budding and internal budding. Okay, in external we will be taking the two examples. One will be of unicellular organism and second one will be of multicellular organism. Under unicellular we will be taking the example of yeast, and under multicellular we will be taking the example of hydra. I will explain internal budding in sponges. Right? Let's define budding first. During budding, a bud or an outgrowth develops from the parent body, and that bud may develop from inside of the body or may be present outside the body. Okay, so that bud or outgrowth develops from the parent body, which can be external bud or internal bud. May be present externally on the surface of the parent body or may be present inside the parent body, right? So that bud which develops from the parent body grows into new organism over a period of time. So let's see how it happens. Let's start with the external budding. In the case of yeast. So how it happens in yeast? As we know that yeast is basically a type of fungus and it is a unicellular structure. It has a unicellular structure. So this is the parent yeast, a rounded body with a nucleus and a cytoplasm. Okay. Now after some time, once the parent yeast gets mature, after the maturity, the parent yeast produce a outgrowth on its surface which looks like a bud. The outgrowth develops externally on surface on the body of a yeast, and this outgrowth receives the cytoplasm from the parent yeast initially. As we see that this is the parent nucleus. Now once the bud is formed or outgrowth is formed, this nucleus will duplicate its content and elongates. Right? So this is a parent nucleus. What will happen to the nucleus? It elongates and gets constricted somewhere from the middle. A constriction appears, right? So the nucleus elongates and constrict. As a result, it gets divided into two nuclei. So one nucleus will remain in the parent body and another nucleus will get into the bud. The second nucleus will get inside the bud. Right. So now the bud is having its own nucleus. It also has a communication of a cytoplasm from the parent cell. Now what will happen after this? Once this bud grow enough, it detaches from the parent body, right? And if it detaches from the parent body, then what will happen is that we are left with two structures. One will be the parent yeast and another one will be the daughter yeast cell. Okay. So at the end of the process, we'll be left with the two structures. We'll be having the parent yeast. This is the parent yeast. And this is the newly formed yeast. Right. Now after this, the newly formed yeast grow as an independent individual or an independent yeast cell. Right. There is a second situation also which is possible in the case of yeast. Instead of the detachment, what happens sometimes that instead of this output, sometimes before this first bud detaches from the parent yeast, a second bud appears on the first one, and before the second detaches from the first, the third appears on the second one, and so on. So this results in the formation of chain of buds. Right? Something like this is going to form, and every structure has cytoplasm and nucleus. So sometimes if the bud does not detach itself from the parent body, then there can be a chance of formation of chain of bud. Right? There can be chances of formation of chain of bud. As the structure resembles the genus Torula, this process of formation of chain of bud is also known as torulation. Why? Because it resembles genus Torula structurally. That's why it is known as torulation sometimes. Right? So let's see what happened here is in the external budding. Why we are calling it as external? Because the bud is formed or the outgrowth is formed externally. Right? So the parent yeast, when it becomes mature, it develops a outgrowth or a bud on its surface. That bud or a protuberance then receives cytoplasm, and nucleus of the parent elongates and constrict so that one nucleus remain inside the parent body, parent yeast, and the second one will be received by the bud. Right? Now what will happen when this bud grows enough? A constriction appears, a cut appears. This constriction divides the parent from the bud, and the bud develops as a new independent organism, though it's small in size initially and later on it grows up. Right? Sometimes the second case happens. In the second case, when the first bud, which is formed over the parent yeast cell, and does not undergo detachment, the second bud develops on the first one, and before the second detaches, the third develops on the second one. Before the third detaches, the fourth develops on the third one. So in this way, the chain of bud is created, and this chain of bud structurally resembles the genus Torula. That's why it is known as torulation. Right? So this is external budding in the unicellular organism, that is yeast. Now we will be talking about the external budding in the multicellular organism. Now let me explain external budding in Hydra. As we know that Hydra is a multicellular organism. It's an animal which belongs to phylum Cnidaria or coelenterate, right? Hydra has a vase shaped structure or body. It has a vase shaped body with tentacles and mouth, right? So these are the tentacles. This is mouth. And this one is the body. Now what happens is, when Hydra undergoes budding, somewhere in the body, multiple number of times cell division takes place. Okay, a rapid cell division starts somewhere in the body part of the body area of the Hydra, right? Due to the excessive cell division, there is no enough space for the cells to remain inside. They start coming out as an outgrowth, right? So they start moving out as an outgrowth like this, right? So these cells, they are moving out and they form an outgrowth which is known as a bud. This is a bud or a protuberance or an outgrowth. Initially, this bud acquires all the nourishment from the parent Hydra, and slowly this bud also acquires tentacles. It grows in size and acquires tentacles and mouth and all the other required or necessary structures inside, right? Once the bud Hydra, it grows fully, right, then it detaches from the parent body with the help of a constriction. The constriction appears here. As a result, this bud, which is a now miniature Hydra, it detaches itself from the parent. It got separated from the parent body. So now, other than the parent Hydra, we have a tiny Hydra, which is nothing but the newly formed Hydra, right? So this is the parent Hydra, and this one is a newly formed detached Hydra, right? And this Hydra grow as an independent organism. Once it detaches from the parent body, it grows as an independent organism, right? So this is about the external budding. Why it is external? Again, the bud is growing outside the body, right? So it's external budding in the Hydra. Initially, due to the continuous cell division inside the body of the Hydra, the outgrowth is formed. This outgrowth, slowly it gets the nourishment from the parent body, and because of the nourishment, this bud acquires the tentacles and the mouth and all the important structures inside the body. And once it grows enough, then it detaches with the help of a cut or a constriction which appears here, and a new independent Hydra is formed. You have seen in both the cases, whether it's a yeast or Hydra, in both the cases, at the end of the process, the parental entity or parental part or the parental organism survives, right? So in both these cases, the parent organism will survive at the end of the process. At the end of the budding, both the yeast as well as the hydra parent will survive. Clear? Now, this is about the external budding. Now we will be talking about the internal budding, that too in sponges. So internal budding is the next subtopic. We are going to talk about internal budding in sponges. As we know that sponges, they belong to phylum Porifera. They mainly exist in the form of colonies. Sponges undergo internal budding specifically when the conditions are unfavorable. During the unfavorable conditions, they undergo internal budding, and the internal bud which is formed in the case of sponges inside their body is known as gamule. This internal bud is known as gamule. Okay? As we know that sponges, they exist in colonies. They have quite a simple body design as compared to other animals. So when the conditions are unfavorable, instead of forming the bud outside, these sponges, they form tiny buds inside their body, right? And these tiny buds are known as gamule, right? So let's talk about the structure of gamule. This gamule, it has a structure as that of a pot, right? So this portion of the gamule, this is internal portion of the gamule. It is filled with large number of homogeneous mass of cells, and these are undifferentiated cells. This is a homogeneous mass of undifferentiated cells which is present in the center of the gamule. These are known as archaeocytes. These are the main asexual body, okay, which actually helps in the asexual reproduction in the case of sponges. So this is about archaeocyte. Then this opening is known as micropyle. As this one is a main structure, as this has to play a main role, so it has to be well protected externally. So it is covered or protected by outer membrane and inner membrane. Moreover, other than that, to provide more protection, this archaeocyte is further protected by the exoskeleton of spicules, these needle-shaped structures which are more or less made up of calcium carbonates and other minerals. So these are known as spicules. Okay, these spicules provide protection. It's an exoskeleton which protects archaeocytes, right? So what happens, as we know that these gamules are formed as an internal bud by sponges, and these are formed during unfavorable conditions. So when the conditions becomes favorable, these archaeocytes, they move out, they release out, they immediately move out of the gamule through micropyle. Okay, all the archaeocytes, the mass of archaeocytes, they start moving out of the gamule through micropyle. When they come out of the micropyle, then each archaeocyte develops into sponge, which later on forms colony. Okay, every single archaeocyte has a tendency to produce an individual sponge, right? So this is how they actually help sponges during unfavorable condition. So what happens in the internal budding? The sponges initially, when they have to undergo budding, because the conditions are not favorable, they produce internal buds which are known as gamule. This is the structure of gamule. Gamule has an opening called micropyle, and it has a very important part at the center, that is archaeocyte. It's a mass of undifferentiated cells which are well protected by the inner and the outer membrane, and there are some spicules which forms an exoskeleton that also protect the archaeocytes during unfavorable conditions. When the conditions become favorable, this mass of cell archaeocytes, they all release out through micropyle, and each archaeocyte develops into sponge which later on convert itself into colonies. Right? So this is about the internal budding in sponges, that is poriferans. So this is about it in today's video. So a quick reminder, don't forget to like and do subscribe to my channel, and please press the bell icon for the latest updates. Thank you for watching my video. Bye.